Welcome. So the goal of this class, which will end at 12:20, by the way, today, so about an hour this time, is to understand that complex networks in biology are made of simple circuits, which we'll call network motifs. So today we'll begin to study our first network motif and how you discover them. And second, this network motif, negative autoregulation, has useful functions. It speeds responses and stabilizes against noise. Makes the circuit robust. So that's our goal for today. So last lecture, we began to talk about gene regulation, and we looked at the dynamics of a single arrow in the network. X arrow Y, which means transcription factor X, binds the promoter region in front of gene Y and changes the production rate of that gene, causing a change in the production rate of protein Y. And remember, proteins are the things that do things in the cell. They are the machines that do all the chemical reactions and build the cell and communicate with other cells and build motors, and they're the composition of the cell that we care about. So we're asking here about the rates of production and the concentrations of these proteins, how the cell decides how much of each protein to make and at what rate. I'm going to make a little digression here to say that we'll have exercises every week, and the first exercise you can download on the Feinberg Graduate School course website. And please hand it in the next lecture, which I guess won't be in the holidays, but the one after. And you can just bring it in a paper form and put it here. Did I explain myself? No. How many people are thinking to do the exercise? Oh, great. All right. So we're talking about gene regulation networks. Many genes with arrows. Arrows mean that this gene makes a protein that binds to a little site in the promoter of this gene, changing its rate of production. It could be a positive or a negative change. This is called activator, this is called repressor. And we looked a little bit at the dynamics. So when I have a gene, this is time, this is Y. I have 0 at the beginning and I start making it at rate beta. Then remember we wrote this equation. Production is beta, and removal, alpha Y, which has to do with active breaking down of the protein and also dilution by cell growth. And when we solve this equation, I want you to show that in the beginning, Y is 0, so this part is very small. So we just have this beta, and you get production. It looks like beta T. You make production at rate beta, and you start like this. But then removal starts to be important because Y is growing. And you reach a steady state. Y steady state, which is the ratio of beta over alpha. Ratio beta over alpha. That's a steady state. And we also asked about the response time, how fast it takes to reach half steady state. Why is this important? Because the steady state is the functional level of that protein. If it's a pump, maybe you need 10,000 copies in E. coli. If it's a transcription factor, maybe you need 200 copies. If it's a ribosomal protein, maybe you need 50,000 copies, etc. So evolution can tune beta over beta and alpha separately, and you can reach whatever steady state. And the halfway point, the response time, T1 half, is the time it takes you to reach 1/2 of the steady state. And the response time turned out to depend only on the removal rate. And let's see if we can understand that. Suppose we make the removal rate very big because we break down that protein. We break it down, break it down, break it down. Right? Then the steady state will drop because beta, if we don't change beta. So we don't change it, so we start with beta and then we end up here. So if we made alpha very big, we'll end up here. But we don't want a low steady state. We want a certain steady state for that protein to function. So we have to, in order to get that protein to reach that steady state, we need to increase alpha. So if we're breaking a lot, we need to be making a lot. We need to be making a lot. And then, so we need to make a big beta. A big beta means a fast production. And then we can reach the same steady state. So this will be beta 1 over alpha 1, designed to reach the same steady state, and it will happen faster. And the amount that you go faster depends on this alpha. If you want to reach a desired steady state, this is a relative measure, right? Halfway to the steady state. Maybe needs a little bit of taking used to, but response time depends only on the removal rate. And as I said before, if you're making and breaking very quickly, and now you stop making, you change very quickly because you're breaking so quickly. And also the same way when you increase production rate. Okay. So why not, if we want fast response times, which is presumably good, if we want to start making a protein, let's make it as quickly as possible, reach that steady state level as quickly as possible. If that's good, why not just increase the removal rate, break all the proteins very quickly and make them very quickly, and then all the proteins will reach their steady state very quickly. And the answer is that that costs resources, because needlessly breaking, that costs energy, ATP, etc. And making also costs energy. So especially making, you need energy to make proteins. And that's why most cells economize. And in fact, they don't actively break their proteins. So in E. coli, the vast majority of proteins, when cells are growing happily exponentially, are stable. And that means for stable proteins, if you recall, this number is equal 1 cell generation. Because for stable proteins, when the cell doubles in volume, concentration goes down by 1/2. And that's the doubling time. That's the response time. So for most proteins in E. coli and also in our cells, they're not destroyed very quickly. And therefore, in growing cells, their response time is a cell generation, which is kind of raises a puzzle, because that means you only respond for your children, basically. Only your children get halfway to steady state. Your grandchildren, 3/4 of the way to steady state. Your great-grandchildren, 7/8 to steady state. So if you want to respond now, you need another solution. And indeed, biology, evolution has discovered another solution, a neat gene circuit that can reach a desired steady state very quickly without needing to break the proteins. And that's what we'll explore today. Did I explain myself? Maybe I pause for a question at this point. And what I mean to say is I'm not going to continue until there's a question. Yeah.
How's beta related to X?
How is beta related to X? So the question is, how is beta related to X? There are these input functions. So if X is an activator, beta increases with X. If X is a repressor, beta decreases with X. And in this exercise, in this I assume that beta is constant over time. So I have some signal, X is whatever it is, beta is whatever it is, and now the protein starts to be made. So we'll soon discuss these input functions, which have beta as a function of X. We'll discuss that. And this arrow is called simple regulation. So it's not a circuit, it's just X activates Y at a constant level, and you have this response time, which is slow. Now, how did we discover this circuit that makes things faster? It started by looking at what biology actually does, by looking at the gene regulation network of E. coli and seeing what's special about that graph. Yeah.
So last time we saw T1/2 equals 1 cell generation for a specific case. Is it generally true?
For stable proteins, that's to say when they're not actively degraded, and most proteins are stable in growing cells. When cells aren't growing, there's a lot of protein turnover. So we're discussing here the case where there's enough food, enough energy, cells are growing exponentially, which is the, let's say, case that we look a lot in the lab. Of course, there's other cases too. So we want to see what is special about the gene regulation network of E. coli. So we're going to use in this course a little well-studied fragment of the E. coli gene regulation network. There's a collection of all the interactions between transcription factors and genes. That maybe in one of the exercises you can download it, and we'll do a little algorithm that looks at graphs, subgraphs in this graph. So you'll get to play with it. So the E. coli gene regulation network, it's about 20% of it. It's very well mapped. So it looks like that. It's a network like this. Okay. It has, in this little part of the network has 400 genes, and it has 500 arrows. These genes are actually operons. They're groups of genes on the same RNA. That's just a detail. So this is a graph. And our approach will be to try to look for subgraphs, as I say, little patterns of nodes and arrows that occur many times in the graph. If they occur many times, maybe they're interesting to look at. So we're going to look for patterns. Patterns are subgraphs that occur often in this GRN. And I want also to look for patterns that occur more often than expected by chance. More than by chance. I'm going to define what I mean. Because maybe any graph you would draw has a certain pattern, and it's maybe nothing to write home about. Okay, so how can we do that? That's a little bit like in statistical mechanics. And our approach will be to compare to a random network. So, or ensemble of random networks that have the same properties. So we look at an ensemble of random networks, and we ask, is there something that occurs in the real graph more than in the random graphs that have the same properties? Did I explain myself? That's really our approach. And if we find a pattern like that, we're going to give it a name. It's going to be we call it a network motif. So let's do that. And we're going to do that systematically. We're going to, in this course, in a couple of lectures, you're going to know the entire structure of this graph, all the overrepresented subgraphs. You're going to, in 2 lectures, be able to tell me the dynamics of the entire big graph, if you stick with me. But now we're going to start just with one, right, to get the hang of it. So we're going to look at the simplest pattern, which has just one node and an arrow going to itself. Later we'll look at 2 nodes, 3 node circuits. This we'll call self-regulation. Biologically, this means we have a gene, and this makes a protein that binds the promoter of itself and changes its own production rate. Mathematically, it means a node with an arrow that comes back to itself. In the real network, there are 40 nodes like that. Is this more or less than expected by chance? So to answer that, we're going to there's different ways to make random patterns. I'm going to make, again, the simplest kind of random network, which is called Erdos-Renyi. Erdos is the very prolific mathematician in the middle of the 20th century, and he also founded this network theory, you can say. And random networks it's a random graph that has the same number of nodes and the same number of arrows as the real graph. So you take 400 nodes, and you're allowed to throw 500 arrows wherever you want randomly. Did I explain myself? By the way, I say, did I explain myself? Because it's my duty that you understand, right? Not, if you don't understand, you have to ask. Yeah. Okay, so you say you can make a random network ensemble that has an average 500 arrows, but let's even make many, many random networks that have 400 nodes and exactly 500 arrows for our exercise now. But now, it's like you say, you can make many choices. You can also keep the number of incoming and outgoing arrows for each node exactly the same as in the graph. You can build very, very elaborate random models. Let's see, so this yeah.
So for the random network, we don't make a difference between activating and inhibiting?
So the question is, do we keep track of the activating or inhibiting signs here, plus or minus? For right now, we won't, but of course you can. You can make a random network that has 200 positive arrows and 200 negative arrows, like this network has, about 50-50. That's right, yeah.
And you're assuming that if you have an arrow coming out of the eye, it has the same probability to go back into eye as to any other location?
That's right. In this very first approximation, we'll assume that if an arrow, so you're already looking at this self-edge, an arrow goes out of a node, it just chooses one of the other nodes, including itself, with equal probability. Right. So in this network, what is the probability of a self-arrow? So if I take an arrow that starts here, it has all these choices, but it chooses itself. It's just 1 over the number of nodes, right? Because it could choose any node, like you said, with equal probability. Again, you can change that. So how many self-edges will there be in this random network? Well, I have 500 arrows, E arrows to play with. And each one has a probability 1 over n to hit itself. So I just throw them. It's like throwing a coin. It has 1 over n probability to end up heads. And to count how many heads, it's just E over n. Okay? How much is that? It's about 500 over 400. It's about 1.2. So on average, I expect 1.2 self-edges. And what's the plus minus on that? So just like throwing a coin, it's the square root of the mean, more or less. So this is about 1 plus minus 1 self-edges in random. And the real network has 40. And yeah?
Didn't we say at the beginning that only a partial group of the edges has outgoing edges?
Yes, so the question is, only transcription factors have outgoing edges, right? And so not every node here can have an edge. So when you take that into account, you get a higher number, but still much lower than 40. Good point. Transcription factors.
Can you explain again what is n-self?
This is number of nodes with an arrow that hits itself. It's n-self. More questions? I just want to say, why compare it to random networks? I mean, for me, every arrow is precious. When you see an arrow in biology, it's already something to study. It's important to recognize that evolution can rewire these arrows very quickly. Very quickly, again, I say on a timescale of hundreds of generations. Okay, so in a given generation, the network is fixed. But hundreds of generations, you have a chance to rewire those arrows. How does that happen? If you get a mutation in this binding site, which is a change of a DNA letter, X won't bind there anymore, or bind much more weakly. And that can happen very quickly. How quickly? If I grow a test tube of bacteria with, let's say, 10 mLs of bacteria. That's how much is 10 mLs? Can I use this? Yeah. This is like 100 ml. Okay. So if I take, let's say, 100 ml of bacteria, like this. And I put a little salt and sugar here, a little bit of bacteria. Within 1 day, I'll have 10 to the 9 per cubic centimeter, 10 to the 11 bacteria here within 1 day. Then they finish their food. Now, the mutation rate is about 10 to the minus 9 in every letter. So I'll have, at the end of the day, 100 examples of a bacteria with a mutation in every possible letter. So for every possible letter, I'll have 100 examples of a bacteria with a mutation there in 1 day. If that bacteria has an advantage, this has no bacteria, by the way, so I'm going to demagic it. If that mutation gives an advantage, right, the next day when those bacteria move to a new test tube, they're going to grow exponentially faster, take over, become a great nation. So what I'm saying is that arrows that have survived this, patterns that have survived this, are probably doing something useful. That's our motivation. And in fact, I want to say that there's a huge probability just to lose an arrow. So every arrow is like a miracle. Okay, so there are bacteria, cousins of E. coli, very close, let's less than 1 million years ago. They, instead of living in us, they live in a termite gut. The termite is very nice to them, supplies them with all the amino acids, all the food, etc. They have lost all their transcription factors. Their genes are produced at a constant rate. They don't need to respond to the environment. So they're very quickly, you can lose all of those arrows if there's no selection for them. They also lost from 4,500, they went down to 800 genes. And you can go down to 400 genes and still be a living organism or below. All right, so why is this pattern a network motif? So let's say we've reached this goal, so now you know what network motifs are, and we'll see in the next couple of lectures that this entire network is actually built of just a small set of network motifs, and we're able to understand the function of all of them, and they're wired together in understandable ways, and it's a big advance in human ability to understand these biological networks. But let's now focus on what might the autoregulation do for E. coli. So I want to say that out of these 34 out of these 40 autoregulations, we're going to call them autoregulation, right? Why autoregulation? Because the gene is regulating itself, are negative autoregulation, which we'll call from now on NAR, negative autoregulation. So I'm going to say that they're like this. X binds its own promoter and reduces its own rate of production. Make sense? Yeah. So guys, what does negative autoregulation do? We, in a good place to take a nice deep sigh of relief. Yeah, and maybe another little interlude as we erase the blackboard. And it's a good time for a question too, yeah. The question is, how many transcription factors are in this group of 400 genes? I don't remember exactly, but I would say it's around 100.
So almost half of them are autoregulated.
Correct. Very often, transcription factors in E. coli and other organisms are autoregulated like that. Sometimes on the transcription level, sometimes post-transcription level, sometimes splicing factors are regulated by splicing, so translation factors are regulated by translation. So there's this. What I'm saying here applies across biological modes of regulation. Let's just check out. It's a gene regulation. Heteroserenity networks. They can be rewired by mutations. Self-edges. Negative autoregulation is a network motif. So let's see what it does. We need to analyze it, and of course what I'm driving at is it can speed the responses and also do other things I'll talk about. So let's try to see how we can analyze that. So negative autoregulation has X reducing its own production rate. So let's do that. So what is the equation? So just like before, we have removal. X is diluted, degraded, etc. But now the production depends on X. So we put here a function f of x, an input function f of x. This input function f of x is a decreasing function of x because x reduces its own production rate. Now, I just want to talk about a little bit about separation of time scales. I mean, how dare I write an equation like that? And here, separation of time scales comes to the rescue because, as I said, X takes seconds to find and bind. And then this production of new X protein takes minutes, much longer. So I can, for a given amount of X, I calculate the production rate. And then X changes slowly, and then there's a new production rate. So I can plug here X concentration because of the separation of time scales. If this part happened as slowly as this part, I will need two differential equations. Things will get much more difficult. For example, we get oscillations and all kind of noise at times. But separation of time scale, very good for us. Just one first order equation. And this happens again and again in biology. Did I explain myself here? Again? Okay. So we need to solve this equation, and you can solve it for any, I mean, you can plug here different forms of x, but we're going to solve it now. We're going to solve it in two different ways, a graphic way and a mathematical way. I'm going to start with just making this approximation a step function, which means very sharp regulation. So this means beta when x is less than some critical concentration k, and 0 when x is bigger than k. Beta when x is less than k, and 0 when x is greater than k. And k, what is k? K is the chemical equilibrium. It's the concentration of x that halfway binds that site, you can say. So it's determined by the structure of the protein, the structure of that site, and it's a chemical thing. It's a very solid number. For every cell, we'll have the same number of k because it's determined by the chemistry of those production. I just want to say in advance, cells have a problem controlling this beta. Cells can vary in their production rate. They're more tired, less tired, have more ribosomes, less ribosomes. But each cell will have the same k because it's determined by the structure of that protein and the site. And a mutation will change k. And a mutation can also change beta, the average beta in the population. Okay, so this is we're going to solve for a step function. And look at what's going to happen here. It's not so difficult to solve. This is time. This is x. And we start with x equals 0, just like we did there. There, I called it y. Yeah, y equals 0 there. And at 0, we're making maximum amounts. We're making beta, right? So beta t. So this is the production rate, beta. And we go up, 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 up. When do we stop? We stop when x goes too high. It crosses k. Then we stop making it, so it goes down. Oh, but then it's below k. Then it goes up. If this function has any little width, it's going to stabilize on k. And maybe if it, I want to say if there's any delays in the system, like if there was no separation of timescales, you can get oscillations here, up, down, up, down. But if there's no delays or small delays, make sense what I'm saying? Yeah.
Can you please explain again the story of the separation of timescales and why it saves this?
The story of separation of timescales. So if if this takes minutes to make and this takes minutes to bind, then there's a delay here. And then you cross k. Oh, but it takes minutes to know. Then you go, okay. And then you need two differential equations. You get like a harmonic oscillator. You can say kind of an oscillator. It's not a harmonic, it's a nonlinear. And then here you can just write one equation because you could put in the steady state value of x. Did I explain myself here? Still not clear?
Not 100% clear, but
okay, maybe it's thought provoking. All right, so now the steady state, what determines the steady state here is this k, this hardwired number, the chemical affinity between x and its binding site. What determines the steady state here? It's the ratio of production and removal rate. You see, removal rate and production rate don't even appear here. It's just this chemical number. So you can tune that to whatever you want by making mutations in the binding site to whatever level you want. So x goes up until it reaches the point where it binds its own promoter and stops itself. What about the response time? So how can I increase the response time here? I can just make beta bigger by making a stronger promoter. If I make beta bigger, so this is T1/2 NAR. And let's just calculate it for the joy of it. T1/2. So, how can we calculate T1/2? In this phase, x goes up like beta t. So beta T1/2 is 1/2 x steady state, which is 1/2 k. So how long does it take me to reach half of k? T1/2 is k over 2 beta. Again, in this case, it doesn't depend on alpha, on the removal rate. It just depends on the production rate. In the simple regulation case, it only depends on the removal rate, not on the production rate. So, for example, if I make beta, making a strong promoter, strong, let's say, RNA polymerase binding site, I can make a beta large and T1/2 as small as I want without need to change alpha, beta, and change alpha, etc. Okay, so here I keep, let's say, a stable protein. It has a removal rate that's very low, but I can still get as quickly as I want to the steady state by making a strong promoter. Strong promoter, bang, right? If I use a car metaphor, I can drive quickly if I have a strong motor and good brakes. Strong motor, and the K, the self-repression is like the brakes. In the simple regulation case, they're linked together. If I have a strong motor, I'm going to go, ah! So I have to make a lot of strong brakes. It's like putting the motor and the brakes together all the time.
What actually happens with K? Is there's degradation and then you make some, and then there's segregation, and then you stop, and then there's segregation and you make some?
What's going on here? So what happens at K? Yeah. So if there's any kind of width here, there always is, then you get, it's actually a monotonic convergence to here. Because of, and I'll soon explain that a little bit better. So
Let's say we started off at K exactly.
at K exactly, right. So
So it can't be constant.
yeah, so if it's a continuous function, then at K exactly, there is a production rate that exactly balances removal. Yeah, and it's stable. It's a stable fixed point. I'll explain that in a second too. So I'm trying to approach these mathematical ideas in different ways. If you're confused now, the next way of explaining will give you another chance to get it. So negative autoregulation can speed responses. Okay, let's do it in another way. And why do it another way? Because you might ask yourself, okay, this is good for a step function, but f of x, maybe we don't know it exactly. Like, as I said, this part is not always known. We just know it's decreasing. Can you prove that this speeds up for any shape of f of x that you can invent as long as it's a decreasing function? So I want to show you a way to prove that graphically without equations. The question about going down, going up, let's look at it. Question of going up and going down. Okay, so I want to introduce another technique. We're doing great with time. So maybe before I do that, make a little musical break. Just again, I say smiling and laughing is good for learning. That's why I do it. So if we're talking about negative feedback, this song is a blues song about negative feedback from reviewers. It's called Dear Authors, we have now heard from 3 referees. It's kind of my music therapy. Dear authors, we have now heard from 3 referees whose comments are attached below. As you will see, they have raised concerns about your interpretation of the facts, your choice of model systems, your references, the style of your prose. Oh yeah. Referee number 1 says the topic is interesting, results are incorrect. Referee number 2 says results are okay, but the topic isn't interesting. Referee number 3 suggests 14 additional experiments, although tangential to your main point would be nice to have. Oh yeah. As a result, we regret that we cannot offer publication at this time. Please know that we value your work and would like to see more of it in the future. At the same time, we remain committed to the high standards of the Archival Journal of Upper Nasal Cavity Research. All right, so speeds, so we claim it's going to speeds response time for any decreasing f of x. And let's see how we can do things and also learn more about these equations. So I'm going to introduce to you a technique called rate analysis. And we're going to start off with simple regulation. And it goes like this. What we plot here, it's a graphic approach. Okay? Here we plot x, the concentration of our protein. So as you recall, dx dt equals beta minus alpha x. This is simple regulation. So we're going to just plot out beta and alpha x. So this is the rates here. So the production rate here, doesn't depend on beta because it's not autoregulation, is beta. And removal is alpha x. And these two lines cross. The crossing point is very important. That's where production equals removal. That's where the rate of change is 0. So this point here, this is x steady state. It's called the fixed point. So the crossing of the rates means production equals removal, no more change. This fixed point is stable. What do I mean? If I start above it, see this point here, removal is above production. So more removal. What does that mean for x? Where does it move? Back. If I started below, production is above removal. More production than removal. Where does it move? So this is a globally stable fixed point. No matter where I start, I end up there. Now the important thing for us now is the speed. What's the speed? The speed, the rate of change, is the difference between the curves. So this distance here between the curves, speed equals the distance between the rates. So as you see, if I'm far away, I go fast and then slow, 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 explaining this fast and slow, 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 slow. Did I explain myself so far? So just by graphically, we can see one fixed point, fast, slow, etc. Now we want to do negative autoregulation. But we want to make a fair comparison. We can't compare, let's say, a really fast circuit to a really slow circuit unless we make sure to give the slow circuit a chance, right? So that's called mathematically controlled comparison. Math-controlled comparison. So we're going to keep as many things as we can the same between the two circuits we want to compare, so we won't fool ourselves, right? This idea was pioneered by Michael Savage-Joe, who was a really important pioneer of systems biology in the 70s. It was ideas from engineering. So how can we do mathematical control comparison? So first I'm going to say this, we're going to have the same removal rate. So if it's a stable protein, it's a stable protein. It's a quickly degraded protein, it's a quickly degraded protein. Same removal rate. Another thing we need, of course, is the same x steady state, because as I said, that's the concentration that we need for that protein. It's the biologically important concentration. And that's the two numbers. We can't keep anything else more fixed. We can't have the same production rate, because that's where the two circuits are different, right? The production rate of one is autoregulated and the other one not. So that means we're going to have the same removal rate, the same fixed point, and now we need to draw the production rate. So how does production rate in a negatively autoregulated circuit look like? It's a decreasing function, because x reduces its own production. That means in order to cross this removal at the same point, the only way we can do that is to start above and go below. This is the only way we can do that. This is negatively autoregulated production f of x. Same steady state means we have to, same removal. Okay. Again, we see very quickly that our fixed point is globally stable, right? What you asked, it's globally stable. No matter where you start, you flow to it, because the production line is above the removal line here and below the removal line here, and therefore you flow. But what about the speed that you move, right? So check it out. The distance between production and removal in NAR, no matter where you are, above or going up or going down, is always bigger because of this decreasing function. So no matter where you are, the distance between these lines is bigger than the distance between these lines. which means faster.
Well, in the simple regulation, you move production to 0 when you want it. Didn't you?
If you want to decrease, you can move production to 0, but you can also decrease by starting above the, fixed point. So if I start above the fixed point, I will decrease like this. And this is what I mean. Yeah, if
Production, and then it's faster.
if it's no production, what you're asking, then negative autoregulation and simple regulation are the same. They're just decay. And in the case of no production, they're basically the same circuits because zero production can't become lower.
No, but
Yeah.
in negative autoregulation, can you also stop production? Because there was either production or no production, if I remember correctly.
In negative autoregulation, you can stop production. Well, production is very low if you have a lot of X because it represses itself. Or there could be another transcription factor there, a very strong repressor that binds. Or an activator is required that unbinds, so you can do that too. And then you just forget about production and everything decays. Yeah. Did I answer your question?
Yeah. I mean, in this situation, yes, but I mean, does the negative autoregulation also allow in some way to actually come from the outside and stop production totally like we did last week?
Yeah, so does negative autoregulation allow stopping production? So here is X
No, but not stopping by it. I mean, just
like this. There could be a Y that binds here. It responds to a signal, SY. I take away the signal, and if this is an AND gate here, or a repressor. Okay, we get to that later. It's an advanced question. So speed, negative autoregulation, is bigger than the speed of simple regulation. And that means whatever happens, you flow quicker to the fixed point, no matter what the shape of this red line is. Did I explain myself? Let's see, let's pause for a question. Or I won't continue until there's a question. Yes?
When you do the mass control comparison, how do you know if it's like the same alpha or the same beta? Because you also said autoregulation can be a negative sign. How do you know what should be the control?
Yeah. So the question is, can you systematically say what to keep constant in mathematically controlled comparison? In this case, it's simple because there's only one possibility to do a mathematical control comparison, which is X steady state and alpha, and beta has to be different because the circuit, that's in the topology of the circuits. But sometimes you don't know because there's different ways to do it, and then you need to try several options. So this idea, I hope, this is a simple case. Sometimes it's more complicated. Yeah.
With the simple step function approximation, when you cross K, so X binds to itself, does it make the degradation big? Because actually you take an X and you remove it.
So the question is, when X binds its own promoter, maybe it's a good time to say hi to our viewers away from this class. When X binds its own promoter, maybe it's protected from being degraded or something like that. Maybe it affects degradation or removal somehow.
No, because it took an X from
Yeah.
the cell to...
Yeah. I can imagine that can happen. That when you take an X away from the cytoplasm, bind it to the DNA, it can affect, let's say, the rate that it's degraded. And that leads to cool mechanisms. You can think what that does. I just want to say that generally, you have hundreds or thousands of copies of this X floating around and just one DNA. So each binding site takes away one copy like that. Sometimes a transcription has 100 genes it's regulating. That takes away, let's say, 100 out of several thousand. So it's a small perturbation, but definitely an interesting circuit. In other cases, like ribosomes, the proteins make up the ribosome, but if they, for some reason, they don't find their friends, they're quickly degraded. And that's a way to control for stoichiometry. If you have too much of a protein, more than its buddies, that need to make a machine in a certain specification of concentration, that you get rid of them. So that is a big effect in some circuits. Listen, so we know that this network motif has a useful function. It speeds response time without carrying the cost of increased degradation and production. And we looked at the rate plot, mathematical control comparison. We did a song. Let's look at another benefit that this has. In order to, this little plot here is going to be very convenient for us because now we can play graphically with other situations that happen in biology and see what these circuits do. So this rate plot is useful for us. Our next topic will be noise resistance. Negative autoregulation makes X steady state robust to natural variations, naturally occurring variations that in fact you can't get rid of in beta and alpha. So what do I mean here? Maybe I should say, well, in production and cell generation time. So here I want to introduce you to the idea that biology, we're dealing with a material called biological material that has a property that you can't evade, which is noise or stochastic nature. It happens on two levels. One is that small molecule numbers. So to make a protein, you copy the DNA into RNA in the process of transcription, and then RNA in the ribosomes, you translate it to a protein. Sometimes the number of RNAs present in the cell at a given moment is very small. It could be 1, it could be 10. And when you try to think about each cell has 10 copies, you have plus minus square root of 10 noise on that. So some cell will have 7, other cell will have 13. Okay? So that means in a lot of things in biology, you just can't, unlike, let's say, engineering, where you can say this resistor will have 100 ohms plus minus 0.5%. You can't do that in biology. You'll have a plus minus 10, 30% variation. Oh, you have to live with it, and still our bodies are so amazing, right? And minds, despite this. So you're trying to make electronics where your capacitors and resistance have a plus minus 30% and still get this precise, amazing body, right? And when I say the word body, 1 million cells in my body die, and another million cells are divide. And if it was a little bit different, my body would grow exponentially or collapse exponentially. So that's pretty impressive, no? How does that work? With these kind of circuits. So we need robustness. We need robustness to noise. Did I explain myself? So robustness to noise is important in cases where we want precision. I just want to say right away, not always we want precision. A very familiar feature of living things is also unpredictability. Otherwise we say it's robotic. So sometimes this noise is used, amplified, to make us different from each other so that we don't all die from the same disease, we don't all get predated by the same predator, we don't... So sometimes you need precision, and we're talking now about robustness to noise. And later we'll talk about using noise as a random number generator, how you do that. So I'm going to use the same plot, and here's X, here's production, here's removal for a simple circuit, simple regulation circuit. Here's the fixed point. Now the problem is that if I look at different cell bacteria under this sitting one next to each other on the same microscope slide, and I use the green fluorescent protein to measure the production rate of a certain protein, I'll find that this production rate beta varies, but as I said, about 30%. That's typical. So you have one cell like this, and its neighbor will have this, higher, let's say. So what happens to the fixed point? It moves. And another one will have lower. And the fixed point, the steady state, moves. And if I plot here the probability to get a certain steady state, I'll have some average, and I'll have a distribution around it. Usually a log normal gamma distribution, which looks like a log normal distribution. Huh? Well, yeah. If alpha also changes, alpha for stable proteins is cell generation time, so it also changes. So alpha. Same problem, right? But now let's think that all the cells do have the same generation time. Okay, so as I said, 30% is typical standard deviation. What's going to happen if I use negative autoregulation? So I'm going to use a kind of a sharp decreasing f of x, like a step function almost, yeah? I want to say f of x varies between cell to cell also, just like beta. But the halfway point, this k here, the halfway point, is hardwired because it's the chemistry of the protein and the binding site. And that doesn't change between cell to cell. It's the one thing that doesn't change between cell to cell. Because it's just determined by the structure of the protein and the sequence of the DNA. So, and what happens is that basically x steady state varies by much less. So you get something like this. And experiments have confirmed that. You make a gene, you fuse it to green fluorescent protein gene, and you make a variety that negatively autoregulates itself, and another variety is exactly the same thing, but you mutate that site. So now it cannot no longer negatively autoregulate. And the experiments show both a speed up. So this is the green fluorescence goes like this or like this, divided by steady state. And variability. So different cells are very different when it's not in simple regulation, but very the same negative autoregulation. So this experimentally has been shown using this ability to take genes from jellyfish, glue them to genes to promoters from E. coli, and measure in the microscope dynamics, etc. So this ability to autoregulate makes you insensitive to changes in beta. There was a question about changes in alpha. So suppose now suddenly there's a little bit more food and the cell grows a little faster. There's a little bit less food, the cell grows a little bit slower. That'll change the removal rate. So in this case, I made removal faster, so it's higher slope. And again, in simple regulation, this will really shift the steady state. But in negative autoregulation, it makes a smaller shift. So that you're insensitive, at least in a certain range, also to changes in cell growth rate. This is very useful for engineers now in this field of synthetic biology, trying to design new circuits inside cells. For example, to make immune cells better at attacking cancer cells and introduce new circuits into them. And these engineers have discovered that when you do that, sometimes you add, you can't control the amount of copies of the little DNA you put into the cells. So some cells 100 copies, some cells 50 that you add. But you don't want the concentration to depend on that, so you put on this negative autoregulation, and you become insensitive to this number of copies of DNA that you put into the circuit, which is like the production rate. And that's very useful technologically, you can say, for designing new circuits inside cells for useful purposes. All this work here on network motifs, the network motifs, I just want to say, was done in my group with Ron Milo, Shai Chenol, Nadav Kashtan, 2002. And the work on negative autoregulation started with Michael Savageau, that I mentioned before, Luis Serrano lab showed around 2002 that negative autoregulation makes the distribution smaller. And Nitzan Rosenfeld in my lab did experiments to show that it speeds regulation in 2002. So this is not very old stuff, right? Next time we meet, we're going to talk about larger circuits that have more than one node and move towards this objective of understanding the entire network in terms of simple building blocks. All right? Time for questions? Yeah.
Can this robustness to noise be demonstrated theoretically?
Can this robustness to noise be demonstrated theoretically? Can you say more? What do you mean?
Well, can you prove from the mathematics that this happens?
Yeah. So the question is, how mathematically do you approach the question of robustness to noise? And of course, one approach is to take the equations and linearize them near the fixed point. I'm going to say some technical stuff, so if you don't understand, it will be a little technical. So we linearize, we change it with a straight line here. And then we look at the eigenvalues, the speeds at which it changes. And then you know that noise and speed are related. So if I change x the faster it goes back to fixed point. Also, noise that kicks it will go back faster. So the bigger the difference between the curves there when you linearize, both the bigger the response time and the better the response to noise. And you can quantify that in the linear regime. And also in the nonlinear case, you can actually prove this gamma distribution. This is work by, for example, Nir Friedman. By the way, all these people are at Weizmann now. Nir is a professor at Weizmann, and Shalev and Ron are now faculty here too. And Shai is at the Technion, and Nadav is in the Hebrew University across the street, and Nitzan is in Cambridge, and Nir is here in immunology. So this, so you can quantify, you can compute this gamma distribution exactly, and compare it to experiments, etc. Did I answer your question? Yeah. There's more stochastic stuff to do. Yeah.
Why are the curves crossing the same point?
Why are the curves crossing the same point? You mean in the presence of noise? Because they don't have to. It's approximately the same point. The halfway curve, as I said, the halfway point, K, is less varying from cell to cell because it depends on the chemistry of the protein binding to its binding site. So the halfway point is about the same. And then the maximum can shift. So if you have a step function, K is exactly the same. Maximum shifts. The fixed point actually doesn't care about beta at all. But if it's slightly not a step function, it will move. But the more slopey it is, the less it will move. Did I answer you? Yeah. Yeah.
Assuming things have a reason, what advantage does the simple regulation have over the self-regulation?
So the question is, if things have an advantage, why do some genes have simple regulation and other genes have negative autoregulation? Great question. So here we come to the idea that biology has multiple tasks. So different genes need different things. Some genes need response time and precision. But other genes, as I said before, actually need variability. And also, some of those edges might participate in larger circuits, like I'll talk about next time, which have their own story. Did I answer your question? So we have multiple objectives.
So typically, how much does it make it more narrow?
How much does it make it more narrow? So typically, in these experiments of it makes it 7 times faster, negative autoregulation, which this particular construct made is a huge. So instead of one cell generation, 1/7 of its cell generation, instead of an hour, 10 minutes. And the noise, there's still residual noise. You can't get the noise down to 0 because there's in fact 2 sources of noise. One is this variation in beta, and the other one is this small molecule noise that you can never get rid of. So you can go down to maybe 10% or 5%, but not below, because additional sources of stochasticity play in. Yeah. This removal line. What I try to portray here is if cells grow faster, and alpha is alpha degradation plus alpha dilution, and let's imagine this is a stable protein. So the removal rate is determined by the growth rate, which is determined by dilution. And so if cells go faster, this whole line shifts here. The fixed point will shift here. If cells go slower, fixed point will shift here. And if you have steep negative autoregulation, you also have shifts, but they're smaller because of this intersection points are moving on a line that's more sloped. Did I explain myself? Yeah. Let's take a nice deep sigh of relief. And I'll see you next lecture. Thank you very much.